substituted just how they would react. 29 and 30 were thought to be possibly charged. And then, yeah, that was those groups. And then our 21 compound, this is also in here. This isn't a compound that we made. It's very similar to ours. It just has the meta substitution instead of the par substitution that you see in our product. Now I'm going to dive into our Ring A analogs and kind of the results that we found with them. Our goals with the Ring A analog, again, is to investigate the effect of various ring substitutions and see how they interact with the mutant EGFR and the Aurora kinase B. Down here, one of our first products that we made is compound 36, which is a chloral substitution, which further in the presentation you'll see is involved in some of our merged compounds. And a newer product that Nick has made recently is compound 40 with a chloral substitution. And once again, these all are on the Ring A, and right here at the 3 position. And here is our SAR analysis. So our Ring 36, which is the first ring, one of the first ones that we made with the 3 chloral substitution, you can see compared to our lead compound, it has really good affinity at the Aurora kinase B that we're looking for, and an increase at the mutant EGFR, 96.7 compared to 83. So that was a really good increase that we saw that made it a good component to be involved in merged compounds going forward. And more recently, the one that I have highlighted is the newer product that we have that has across the board really good numbers for inhibition at all of the receptors. And then going into our Ring B analogs were important in our research because we were trying to optimize the allosteric pocket binding and inhibition. The goal here was to, once again, investigate the effects of different groups. And then our lead compound, Ring A here, with the 3 position being constant. We did several substitutions, as you can see, 3,4,5 trimethoxybenzalamine. On the next slide I'll show was one of them that actually showed really good results. And here's our activity chart for that. Our lead molecule is at the top, so it gives us kind of like a base to compare to. And we're looking for our Aurora kinase B inhibition is really good, but we're looking for an increase in the mutant EGFR inhibition. And if you look at 30 and 30D here, which is our 3,4,5 trimethoxybenzalamine, we had a really good display of inhibition there, which also made that a really good component for merged compounds going forward. And here is our merged compounds, Ring B A analog. So with both substituents there, once again, investigating how both substitutes will play a role. And we have our compounds here, and our 3 substitutions, the 4 that we have made. 3 substitution is mainly made of the chlorine substitution that I showed you guys on the Ring A SAR analysis. And then we have different 4 substitutions for Ring A right here. These are still waiting to get SAR analysis, so we're looking to update that as well. And just an overview of the SAR analysis. The SAR analysis provided us a percent inhibition of Aurora kinase A, B, and the mutant EGFR in comparison to our lead molecule. Our Ring A analogs, 36 and 40, both demonstrated great inhibition of Aurora kinase B and the mutant EGFR. But our newer compound, 40, provided improved inhibition at the Aurora kinase A. That was the fluorine substituted at the 3 position on Ring A. And then 30D, which was the 3,4,5 trimethoxybenzalamine, it showed good inhibition at the mutant EGFR compared to our lead molecule.
So just wrapping up with what comes next on this work. So we'll take the molecules that appear to perform well on those inhibition studies, and we'll perform enzymatic assays to get IC50 values. This will be closer to being clinically relevant in designing drug candidates versus just the percent inhibition would be. We'll also look at how they perform in non-small cell lung cancer cells. That'll be in collaboration with the AMISA lab. And then there are other substitutions we'd still like to try at both ring A and ring B positions, just to continue to optimize those positions and see if there's still better compounds we can make yet. And then once we've optimized those positions, we'll continue to explore different sites on the prolopyrimidine scaffold, such as these locations, to see if we can even further improve specificity and inhibition of our targets.
And then there's a picture of us. We're just going to talk briefly about what got us into research. I started in the lab at Grand Valley. I was involved in like organic chemistry labs and microbiology labs and really liked being in like a hands-on environment. And when I came here to Ferris, they had an elective for a lab. I was fortunate enough to get involved in Dr. Kurup's lab, and that led to an assistant position over the summer and getting to continue to do what I like to do. This really has kind of helped me throughout all of my IPs because it gives us kind of a different perspective on a lot of the ways that drugs work. Instead of just focusing on like side effects and mechanisms of actions, we can kind of look at medications and get a good understanding of what molecules are doing actually. And my goals going forward are going to be to make it through my P4 year and hopefully continue to be involved in research like this.
And so I did my undergraduate years at Grand Valley. I got a degree in biochemistry there before I started here at Ferris. And I sort of regretted not getting involved with research there. I tried to, but it was a little too late. So I kind of missed that opportunity. So when that presented itself here to join Dr. Kurup's lab, I made sure that I took it this time. And for my career path, I'm currently interested in a clinical oncology role. So the fact that the lab was focused on oncologic subjects was just a benefit there. So, and we just like to finish with some acknowledgement. First with Dr. Kurup and the other Kurup lab workers who helped us get everything to the point it's at now. The folks over at the Shimatsu Core Lab, both faculty and student workers who helped us with analysis and the use of the instruments over there. To the university and the College of Pharmacy for making this opportunity possible for us, and also to our financial support there. With that, we'd be happy to take any questions you might have.
Structurally, how many, I don't know how to ask this. So you're looking at an ATP binding site that is common to both the or enzymes, kinases, and the PGRF. How many other enzymes potentially have that same ATP binding structure that you might potentially interfere with? Have you investigated that?
Sure. So just to make sure everyone was able to hear, the question was, to what degree might our molecules interact with other ATP binding pockets? And the answer to that is that's absolutely a possibility, and that's a challenge that needs to be addressed. So like I mentioned, there's a really high degree of similarity between ATP binding pockets and different kinases. They all recognize the same thing. And there are a lot of kinases that we all have that perform incredibly important functions. So creating specificity toward your target is a challenge that needs to be addressed. And that's really one of the things we're looking at doing here, is seeing if adding substitutions that go on that allosteric site can negate hitting some other kinases and really hone in on the aurora kinases and this mutant EGFR. Some of the EGFR inhibitors that have been marketed, afatinib is an example. It hit both EGFR, the mutant one you want, and all the wild type EGFR that you don't want. And that caused a lot of side effects, rashes, diarrhea that was pretty limiting from a clinical perspective. So it's a challenge that is going to have to continue to be addressed because there's huge potential for it. But with proper allosteric site modifications, we hope that we can really tailor it to the kinases that we're looking for.
I would like to add to that. I'm not sure about the research. So the lead molecules that we start off with, we've tested that against the 96 guidance panel. and it is selective for TGFR, aurora kinase, and 2 other kinases. So we try to treat that out. Since their molecules are based on something that we know is selective, we're hoping that their compound remains selective. So once we hit something in a better range, we'll do the selective testing. Okay.
Thank you.
All right. Can you hear?
We have one person.
Question. Can you hear me?
Yes, Dr. Misa, we hear you.
Yeah, I'm sorry, I'm driving, so there's a little bit of background noise. So good job. I think I'm impressed with your presentation. I've really enjoyed it. Now, my question is on one of your slides, and I don't know whether you already answered it already. So you have percentage inhibition of the various enzymes that you were testing. Now, how does that translate to the level of specificity? That's my first question. And the second question is, how specific do you want your compounds to be, knowing that inhibition of other kinases downstream the RAS signaling pathway could be of an advantage? So my first question is, yeah, this slide, you gave a good presentation on the percentage inhibition, some of which are quite high. Now, how does that translate to specificity? Because you want to be as specific as possible regarding the target. And much as you want to be specific, that comes in with my second question. How far do you want to go? Since if you're able to inhibit some of the kinases downstream RAS, like RAF, MEK, ERK, you would also offer some advantage towards killing the tumor cells.
No, we'll give it a tap. Do you wanna start?
Yeah, sure. So from my understanding with the SAR analysis, we're looking for a percentage inhibition greater than 90, and our main targets for our products are aurora kinase B and the mutant EGFR. So what we were looking at is comparing it to our lead molecule, and one of the things that our lead molecule had that we didn't necessarily like was the percent inhibition of 83 on the mutant EGFR. So we were looking for our compounds to have a greater value than 90 for aurora kinase B and mutant EGFR.
Yeah, and Dr. Misa, I'll expand on that. So a high percent inhibition of our targets is sort of the bare minimum we would look for, and even considering one of these to be a good candidate. From my understanding, it's not really going to give us information regarding specificity. That's something that's going to have to be assessed separately. So I view the high percent inhibition as really the bare minimum that we would expect from a good candidate. And then in terms of inhibiting other downstream kinases that may be relevant, that would be great. I think if you can tailor a molecule to hit all of the oncogenic kinases, then that would be, the silver bullet to taking care of cancer. But I think that would be a real challenge to continue to hit only the kinases you want and not any others. I'm not saying it's impossible, but it's probably somewhat close to that.
All right, that's excellent explanation. I'm looking forward to testing your compound, but please, if you realize that there's an off target to MEK or ERK, don't throw it away. Thank you very much.
I would like to add to that.
Yeah.
I think you answered it very, very well. For now, our criteria is anything above 90 is what we are focusing on at the moment. When we say selective, anything that's less than 50%, we're moving out. So if it's not inhibiting any of the targets at 50% or less, then we're not considering that to be a hit. And then for Dr. Misa, once we get data from you on the cell work, or once you have, once we give you the compounds to test, if we have something that inhibits cells, that is not what we were expecting from our data, then we will probe to see if it hits any other kinase. So that way we can figure out if it hits any other target.
Okay.
That's the probe. Those are the steps we're going to follow as we identify.
Okay. Thank you.
All right.
Well, thank you for your
Thank you,
time.
everybody.
Camera. Oh, it's that one? So that one is doing the 3 degree? Not 360? Oh, you're right. Never mind. It felt like it was 360. Sorry. I'm in 360, aren't I? You want to go read the room? I know they have the owl, and it doesn't look like the owl.
This is good. Okay.
Nice.
Nice
Yep.
job, guys. Thank you very much.
I did your presentation right on the desktop. We tend to run better, not from. Online.
Okay.
Poultry. Yeah. I thought maybe because help you find things. So that's by finding this thinking that you come to I know. Yeah. Yeah. That was my exact question. ATP binding. I mean, not even just the kinase, right? But so many binding sites. So forgetting. But innovation.
Here's your mic.
Right.
Pretty much this whole room.
Yeah, exactly. Yeah.
All right, we're going to begin our second session. And we have Dayna Gesinski, if I said that correctly. Very good. She's going to talk to us about microwave chemistry for anti-cancer drug synthesis. The floor is yours.
Thank you. Hi, everyone. My name is Dayna Gesinski. I'm in my third year of undergrad here at Ferris in the College of Arts and Science. I'll be starting my first year of pharmacy school this fall. I've had the privilege of working with Dr. Kurup for about the past year, and the majority of my research has focused on microwave chemistry for anti-cancer drug synthesis. I don't have a lot of background because most of it is the same as Nick and Justin's background, but the compound shown there is a compound that we found was a dual inhibitor for EGFR and aurora kinase B. It's ATP competitive and allosteric, and it doesn't inhibit aurora kinase A. Majority of my research has focused on substitutions at the 6th position. The 3 compounds shown there are compounds with amine linkage, which allows for additional hydrogen bonding within the pocket. And the 6 substitutions allow us to explore additional interactions in the ATP pocket. This is the scheme for my reaction. I started with a 4-fluoro-6-carboxylic acid pyrroloprimidine and ran a displacement reaction with 4-benzyloxyaniline, isopropanol, and concentrated hydrochloric acid. Reflux that for 15 hours to get the intermediate. And then ran the second reaction with amines, PIBOB, DIEA, and DMF for 72 hours at room temperature. These are the 3 types of 6 substitutions that I've work with. Compounds 5 through 8 have a sulfur linkage, which allows for different orientations at the binding pocket. The 6 substituted alkyne and the 6 substituted ether linkage in compounds 9 through 13 and 14 through 18 allow for different orientations in the binding pocket as well. Microwave chemistry is used for reduced reaction times, higher product yield, and cleaner results. The 2 types of microwave chemistry I've worked with are Sonographer reactions and Ullmann reactions. Sonographer reactions are used to form a carbon-carbon bond with an alkyne, and it uses copper and palladium catalysts for the reaction. And Ullmann reactions are used for coupling to air halides. I've used thiols for my reactions. This is the scheme for the Ullmann coupling reactions I've done. I started with a 4-chloro-6-iodo-pyrrolopyrimidine and combined that with 4-benzyloxyaniline, isopropanol, and concentrated hydrochloric acid. Reflux for 15 hours to give compound 20, and then use a thiol with copper iodide, potassium carbonate, and palladium in the microwave for 3 hours. This is the Sonogashira coupling reaction. I started with the same starting material, the 4-chloro-6-iodo-pyrrolopyrimidine. Same reagents to make compound 20. And then from there, ran a Sonogashira reaction. Palladium, copper iodide, DIA, DMF, and that reaction took 10 minutes. The techniques I've learned while working in lab have done a lot of reaction setups with both microwave reactor and in the fume hood. I've done compound purification with the ComboFlash chromatography system. Nick has done mass spec for our group, and we've also had NMR to confirm the compounds that we made. These are some of the instruments in lab. At the top left is the monowave that I've run the Sonogashira and the Ullmann reactions in. The middle picture is of the hood we use, and the top right is for the rotovap, which we use to make silica plugs of our company. These are the results when the compounds I made were taken in and tested for percent inhibition of aurora kinase and EGFR. Compound 3 performed well and had a 97.3% inhibition against aurora kinase A, had a 99.15% inhibition against aurora kinase B, and a 99.5% inhibition against EGFR. Compounds 15 and 16 also the high percent inhibition against EGFR. In the future, I'm looking to complete the synthesis of analogs of compound 1 to see any differences in activities. Also looking to evaluate those compounds against EGFR and aurora kinase, and we'll perform a cellular evaluation in non-small cell lung cancer cells in collaboration with the AMISA lab. And I'd like to thank the other members of Dr. Krup's lab, and of course Dr. Krupp, NMR spectroscopy at Grand Valley, the Ferris State College of Pharmacy, and Ferris State University, and of course our funding. Thank you. Answer any questions.
Can you explain the chemistry of how the microwave actually helps
reaction?
I believe it was reaction type on the slides where you're showing the different compounds. Where you hit some steps. So, not this one, one before, that one. Where you would microwave. Can you explain how that actually, how that works, how to speed that up?
That's something I'd have to look into.
All right, thank you.
But I can explain if needed. So the solvents play the role, and then it helps align the dipoles of the molecules in the same direction. So that's basically the base of the reaction.
Okay.
But the extent of
Can
what I know.
you do the same thing without microwave and just do it longer or something? Or is it
Yes.
something?
No, it
okay.
can be done, but it would take much longer.
Okay.
So the microwave actually shortens reaction time.
Does the ring structure of the compound help at all with the microwave
I guess, yes.
to capture that,
Yes.
some of that energy?
I'm not exactly sure of that. I'll have to look that out. But I would say just the dipoles. And I guess maybe that's true, but I would say just the dipoles get aligned.
Okay.
So how much does it expedite the process to use the microwave?
So for the Sonographer, it takes normally overnight reactions to get it to go somewhere around 14 hours. I haven't had Dayna do any of the non-microwave ones. So she ran it only in the microwave for 14 hours, it goes down to 10 minutes. So that's the
Wow.
yeah.
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 And that can be accommodated with the undergraduate course schedule.
Right.
So that's why we used it, and it's worked for us. We've not got some to work, we're still trying to develop methods for that. For the almond coupling, those work, for some reason, don't work as well outside the microwave at all. Whereas here they kind of put it in the so that's another distinction. But even in the microwave, if you look at the time, 3 hours is still a significant amount of time, So
Is that like a new area of chemistry, microwave chemistry? Has it been around forever?
I would say for the past decade, it's been used. It's not, it is included in some labs. It's not as new, but it's not as routine either.
Okay.
Is it optimized for the wavelength that the different wavelengths make?
No, it's just, well, I guess you just get devices. You don't have to really change anything with respect to the wavelength or anything. It's just an automated device. Just put in the temperature you need and the duration of time.
Okay,
Like
wow,
a microwave.
wow. That's really cool, actually.
Yeah.
The only thing is, it's not these high pressure settings. So like a typical microwave that we have at home, it can't contain an explosion.
But still no metal going in there.
Yeah. All of that.
Oh, I noticed that you've got 2 grants, so congratulations on that.
Thank you. It's all because of the students, it's all the preliminary data that they generate. All of these compounds that we find. Very nice and enthusiastic students, really.
Congratulations. So I saw one was in the NIH, I'm sorry, I just put the second one in.
The other one's to the NEC, the advanced grant for proof of concept. So I got the advanced first, that helped us get some data. That's, I think, before that, when Nick joined our lab, or I don't know if it was Nick or Adrian, one of them. And then after that, it went to, that led to the preliminary data at the NIH. So, it helps to have students, they really do all of the work that's needed. So
The first one's through NEC, which is what?
state funded, so Michigan, education, development.
Okay. Awesome work.
Thank you. Well,
Everybody.
Nice job, Dayna.
So we got a little relay. So, Cliff is coming.
Yeah, we got 18 minutes.
No commute.
You know what?
No room to commute.
Yeah. No commute, just sit and stay. I'll take the notes. Okay. Oh, okay. Okay. Yeah.
I'm so proud of you.
Yeah, let's start. Pretty nice. Did you hear this? So I think what Bobby did today when he thanked us, he did at the thing as well. Because when I came late, remember?
Yeah.
So I went, started at 4 to 6 and then
I would talk to
Um,
her probably.
we spoke early. I didn't get there till after 5:30, 5:20. But I guess Mike Berghoff was that day. I'm very active. I'm like,
There was
what? I'm like, what did I forgot about our 5 M's.
Yeah.
I'm like, oh, yeah. He said 5 yesterday, so. For the wastewater. Those are, actually, we didn't even talk about, well, we mentioned that we went to the conference in here, but we didn't mention, I suppose we could say that. 5 different. Oh, we did that. Yeah. So, you know, so the president was sitting next to me. You know why he came and sat next to me? He goes,